Dear students, today we are going to start another chapter, that is translation, and till the last chapter we finish all the detailed aspect of transcription. What is translation? Its simple definition is known to everybody. I hope like that. What is translation? Translation is the decoding of nucleotide sequence that is present in mRNA to amino acid sequence in a polypeptide chain. Okay, the sequence, nucleotide sequence in an mRNA, it is decoding to amino acid sequence of a polypeptide chain, and that will later form a part of a protein. Okay, so that process is simply translation. That is what is playing here. You can see that from DNA, mRNA is forming, then mRNA, then translator to a polypeptide chain. Later it become one part of a protein. Okay. Properties of genetic codon. Codon is a triplet codon. How? How we all came to know that the codon that is corresponding to a particular amino acid composed of 3 letters. How we came to know that? And that triplet form of each codon is proposed by George Gamow. And that conclusion as a result of an armchair logic. Armchair logic means it is a simple logic of this person, George Gamow, while he is sitting in an armchair. So it's just an outcome of the way how he think about the codon. So we are going to understand how that idea comes out. Now only 4 bases are available. We know that which all those bases. They are adenine, guanine, cytosine and thymine. All these 4 bases are available to form codons. And first he think that if the codon is composed of only one letter, then how many codons can form? There are 4 letters. So 4 raised to 1 is equal to 4. Only 4 codons is possible. And using that 4 codons, only 4 amino acids we can represent. Actually, in our body, 20 amino acids are present. So, it is not possible. So, then he think about codons having 2 letters. And 2 letters means 2 combination like A, G or C, T like that. So, in that way, how many possible codons? 4 raised to 2 is equal to only 16 codons only possible. Oh, that's also is not enough. Then, what next? Then he think about each codon is with 3 letters, 3 bases. With that way, how many codons is possible? Yes, 64 codons are possible, and this is more than enough to represent the available 20 amino acids. So in that way, he came to the conclusion that each codon that is representing amino acid is composed of 3 letters, 3 bases. Okay, and these are all bases attached with the nucleotides in mRNA. So, 3 bases, 3 nucleotides constitute for 1 amino acid. Okay, this constitute for 1 amino acid. Clear? Okay. In that way, it was found out that codon consists of 3 letters or 3 bases. Then next doubt is that whether that codons are overlapping or not. Overlapping means what? I will show you. Overlapping means Suppose this is one codon AUG, then CTG. Okay, then this will code for one amino acid. Then next UGC code for another amino acid. Then again GCT code for another, then CTG again for another amino acid. This is overlapping. This is the doubt whether these codons are overlapping or not, and that doubt is clear by studying the case of sickle cell anemia. What is the case of sickle cell anemia? See, in sickle cell anemia, that diseased condition happened because of the change of glutamine amino acid to valine. Normally, glutamine should be present in the sixth position in the polypeptide chain, and that glutamine, single amino acid, is changed to another amino acid. So again we can write that the codons AUG, they are actually not the codon corresponding to the glutamine. I am simply writing some codons. CTA. If the codons are overlapping, if a change happened to a single base, suppose AUC, then CTA, then what will result if the codon is overlapping? Then this will form one amino acid. Again, CTC form another amino acid. Then CCT form another amino acid. In that way. Okay. uh, Normally, otherwise, if that codon is not an overlapping, again we can rewrite the same changed sequence once again, and consider that if the codon is not overlapping, then this will code for one amino acid, this will code for one amino acid. Okay, that means that change in one base results in the change in only one amino acid. That only happens here as a result of changing one base. Only one amino acid is changed. If that codon is an overlapping codon, it may result in three different amino acids, but that is not happening in the case as the scientists studied the case of sickle cell and anemia. So that study underlined the fact that the codons are not overlapping. It is, it is separate itself. Three codons as such is representing one amino acid. Okay. Next is about the degeneracy. Codons are, there is a redundancy or abundance in codons is there. Whether it is correct or not. It's correct. Because we know that with three bases, 64 codons are possible. And in our body only 20 amino acids are present. Then there will be some excess codons. What these codons code for? We can take an example AUG, then again AUC, then again AUT. All these codon will code for a single amino acid. That means what is the base in the third position is not that much significant to code for a amino acid, code for an amino acid. Only the first two bases are important. That means a single amino acid can code by more than one codons. So a degeneracy is there. If somehow a mutation happened to a codon, another codon can take its responsibility and can synthesize the corresponding amino acid. Okay. Next is identifying the codon. So till now we have learned that there will be a 3 base codon to code for each amino acid. Then we need to understand what amino acid is coded by each type of codon. Okay. For understanding that, two scientists, that are Marshall Nirenberg and Heinrich Mathai, they synthesize an artificial mRNA, and that mRNA is having a polyuridine sequence. Polyuridine sequence means it is that mRNA sequence is simply multiple repeated sequences of uridine. That is that mRNA, and they found what protein is generated, synthesis from this mRNA. And that experiment I will show you. This is that mRNA, artificial mRNA they generated or synthesized to test or to study about the codons, and then they introduced that mRNA to a test tube, and in that test tube it contained all the 20 different amino acids and all the ribosomes that are necessary for performing the translation. And they wait for some day. After some time, from that mRNA, which polypeptide form a polyphenylalanine is formed after some day. That means that UUU codon code for the amino acid phenylalanine. Since there are more UUU codons, that is why a polyphenylalanine polypeptide was formed there. Okay, that is that experiment, and in that way they understand that UUU, that codon is corresponding to the amino acid phenylalanine. Okay, next we are going to see about the universality of the codon. If we take a cell and consider each codon, the amino acid that is encoded by each codon should be universal. It should not change from one cell to another. If AUG that codes for a particular amino acid, then that same amino acid should be coded by that particular codon in all other cells also. It should not change. It should be the same. That's what universality. But at the same time, that same codon, I will make it more clear. Suppose UGA, say codon, and the nucleolar mRNA. That means the mRNA formed from the nuclear genes. This codon is a stop codon. Stop codon means in the translation process, there are certain points where the translation need to stop, and that pause points are indicated by these colon. Wherever or which all places, if a particular mRNA have this sequence, when that ribosome and all the machinery for the translation reach that particular UGA sequence, then it recognizes that this is the point to stop the translation, and the translation is stopped. So that's why this codon is called a stop codons. So that is universal in all cell, all body cells corresponding with respect to that nucleolar mRNA. UGA is the stop codon. Coming to the case of mitochondria, we have learned that we know that inside the mitochondria also DNA is present, and some of the amino acid required for the mitochondria is synthesized by the mitochondrial genome itself. So there also transcription and translation is happening. Inside the mitochondrial genome, mitochondrial mRNA, that UGA codon, we know that we understand that UGA is a stop codon for all nucleolar mRNA. But for this mitochondrial mRNA, that UGA corresponds to an amino acid, not a stop codon, tryptophan. And also, for three other differences exist with respect to the codon in the case of mRNA and normal nuclear mRNA. What are the other differences? AUG is another codon. And in nuclear mRNA, AUG is the codon corresponding to an amino acid isoleucine. Isoleucine. But in mitochondria, this codon corresponding to the amino acid methionine, and AGA and AGG. These are the codons that act as stop codons in the case of mitochondria. But coming to nuclear mRNA, these codons are corresponding to the amino acid arginine. So these are the differences. Even if the rule or the amino acid that need to be coded by each codon is universal to all cell. There are some exceptions when coming to the case of mitochondrial mRNA. That's what I explained here. Okay. Next is about the safeguard aspect of codon. Regarding this point, I just mentioned some points in the former slides. Again, I will explain that to you to make it more clear, the safeguard aspect of codon. I will draw a polypeptide. This is a polypeptide with subunits of different amino acids, like that. And the amino acids that are having similar properties are clustered in a particular area. That indicates that the codon, the corresponding to this amino acid in the mRNA, suppose AUG, we can write it, AUG. That codon that is coding that amino acid. I already told you that there are 64 codons are available to code for 20 amino acid. Okay, so that means for representing a particular amino acid, there are more number of codons. Okay, so AUG, then again AUC, then AUT, and all these codons can code the same amino acids. Okay, so that's why the amino acids having similar properties are clustered together. And if somehow a mutation happened and a base changed in one of the codon, then what will happen? Again, that same amino acid will be coded by this codon. Why? That also I explained to you in the former slide. Because the third base is not that much significant. The determinancy of that amino acid is mainly done by the first two bases. So that's why even if a mutation happened, then also the sequence of the polypeptide chain will not change. Okay, that's a safeguard aspect. As these codons corresponding to similar amino acids are clustered, arranged together in the mRNA, then that is why that amino acids having similar properties clustered together in the polypeptide chain also. And as these codons are arranged in same location, somehow if any mutation happen, then also the arrangement of the amino acid sequence will not change. If that sequence and that order of amino acid change, then that will affect the property of that polypeptide. Okay, is it clear? Next is decoding the codons. Decoding, we are starting the decoding. The codons that is present in the mRNA going to decode to amino acid sequence. And the main adapter, that is the main agent that is helping for that, are tRNA. tRNA are the RNAs that are carrying the that specific amino acid to a particular complementary codon that present in the mRNA. Okay, is it clear? mRNA contains codons, and corresponding to that, a particular amino acid need to be brought to here by a tRNA. Here that amino acid is carried tRNA, and that tRNA need to make an interaction with the codon that is present in the mRNA. This codons, it has a name that we learned in the chapter that you study about structure of tRNA. So the codon that present here are called anticodon. So that anticodon interact with the codon of the mRNA. That should be complementary. As here is A, here should be U. As U, there should be A. As here G, and here it should be C. So that is that interaction. And that interaction add the specific correct amino acid to that position. Okay, so decoding is done by tRNA. They act as adapters. Each tRNA is linked to a specific amino acid. How that linking, that I will explain to you later in the coming slides. Then that tRNA recognizes a particular codon in the mRNA by interaction between successive codon in the mRNA and specific amino acid tRNA using its anticodon. Okay.
Next, we are moving to an important section, that is Wobble hypothesis. That's why I'm marking it as very, very important. Who proposed this Wobble hypothesis? Francis Crick. He proposed Wobble hypothesis. And this Wobble hypothesis is an explanation for the degeneracy of codon. What is degeneracy of codon? That we have seen in the former slides. That means multiple codon can code for same amino acid. That is degeneracy or redundancy of codon. In the case of Wobble hypothesis, or the Wobble hypothesis says that the base pairing between anticodon and codon, in that case, the first two bases, they follow the Watson and Crick rule of base pairing. Only the first two bases of anticodon and codon, they follow Watson and Crick rule of base pairing. And the third one, it will not follow Watson and Crick rule of base pairing. Instead, it have a Wobble pairing. Wobble pairing means unsteady pairing. So I think that all of you know what is the Watson and Crick rule of base pairing. Anyway, just to brush up your memory, I am just writing here what is the Watson and Crick rule of base pairing. According to that rule, adenine always pairs with the uracil or thymine, and guanine always pairs with the cytosine. This is the Watson and Crick rule of base pairing. As we are talking about tRNA only here, we don't need to consider about this thymine. Only need to talk about this uracil, adenine, guanine, and cytosine. Okay, next I am going to show a simple representation of Wobble hypothesis to make the idea more clear to you. So let's see. So this is one mRNA containing different codons. Look to these codons. All these codons have first two bases. All the codons have same first two bases. That means all these codons are meant for the same type of amino acid. Okay, clear. So, I'm just writing here the Watson and Crick rule of base pairing. Adenine base pair with the uracil and guanine base pair with cytosine. This is Watson and Crick rule. If it pairs like in this way, if adenine pairs with the, sorry, we can take a different color. If the adenine pairs with the cytosine, then it will become a Wobble pair. And if the guanine pairs with the uracil, then it will become a Wobble base pair. And there will be one more unusual base, that is inosine or hypoxanthine. It is a naturally occurring purine derivative. Naturally occurring purine derivative. And this inosine can form Wobble base pairing with which all bases? It can form Wobble base pairing with adenine, cytosine, and uracil. So let's see what is happening. So first came one tRNA, and the first two bases between anticodon of this tRNA and codon of this mRNA follows the Watson and Crick rule itself. Adenine base pairs with uracil and uracil base pairs with adenine. And this guanine. And guanine also here follows the same Watson and Crick rule. Anyway, it form a base pairing with this codon of this mRNA. And after that, the same tRNA can base pair with another, the second codon present in the mRNA. The difference is that the first two bases, they are following the Watson and Crick rule of base pairing. But coming to the third base pair, G, according to Watson and Crick rule, it need to base pair with C, but instead it is forming a base pair with the uracil. That is a Wobble base pairing. Okay, and there come another tRNA. Another tRNA means here again the first two bases, they follow the Watson and Crick rule of base pairing itself. But the third one is inosine or hypoxanthine. I already told you that it always form a Wobble base pair with with other bases like adenine, cytosine, and uracil. So all these base pairing, it can form base pair with this codon and this codon. All these base pairings are Wobble base pairing. Okay. Then came another tRNA. Look, it's anticodon sequence, base sequence. Here also, first two codons follows the Watson and Crick rule. But the third one, instead of forming a base pair with uracil, it form base pair with this cytosine. Again, a Wobble base pairing. Okay, I think the idea is clear to you. Here, 1, 2, 3, 4, 5, 6 codons are there. But for the 6 codons, only 3 tRNA are required to form base pairing to encode the corresponding amino acid to the polypeptide chain. So, it supports the idea of degeneracy of codon. Okay, so let's see what's the definition for Wobble hypothesis. The Wobble hypothesis proposes that the third base of a codon and the anticodon of the tRNA can exhibit flexibility or wobble in their base pairing, and that allows a single tRNA molecule to recognize and bind multiple codons, contribute or substantiate the idea of degeneracy of the genetic.
Okay, with this we finish today's class and section. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in the Benz.